Welcome to IAOCS.com Food Tech Club. Today's topic is processing methods of frozen fruits and vegetables. Freezing is a unit operation in which the temperature of a food is reduced below its freezing point and a proportion of the water undergoes a change in state to form ice crystals. The immobilization of water to ice and the resulting concentration of dissolved solutes in unfrozen water lower the water activity of the food. During freezing, sensible heat is first removed to lower the temperature of a food to the freezing point. In fresh foods, heat produced by respiration is also removed. This is termed the heat load and is important in determining the correct size of the freezing equipment for a particular production rate. Energy of freezing is supplied as electrical energy, which is used to compress gases, that is refrigerants, in mechanical freezing equipment, or to compress and cool cryogens. If the temperature is monitored at the thermal center of a food, that is the point that cools most slowly as heat is removed, a characteristic curve is obtained. The six components of the curve is as like the first one, AS. The food is cooled to below its freezing point F, which, with exception of pure water, is always below 0 degrees Celsius. At the point S, the water remains liquid, although the temperature is below the freezing point. This phenomenon is known as supercooling and may be as much as 10 degrees Celsius below the freezing point. SB, the temperature rises rapidly to the freezing point as ice crystals begin to form and latent heat of crystallization is released. BC, heat is removed from the food at the same rate as before, but it is latent heat being removed as ice forms and the temperature therefore remains almost constant. The freezing point is gradually depressed by the increase in solute concentration in the unfrozen liquor, and the temperature therefore falls slightly. It is during this stage that the major part of the ice is formed. CD, one of the solutes becomes supersaturated and crystallizes out. The latent heat of crystallization is released and the temperature rises to the eutectic temperature for that solute. DE, crystallization of water and solutes continues. The total time TF taken is determined by the rate at which heat is removed. EF, the temperature of the ice-water mixture falls to the temperature of the freezer. A proportion of the water remains unfrozen at the temperatures used in commercial freezing. The amount depends on the type and composition of the food and the temperature of storage. For example, at a storage temperature of 20 degrees Celsius, the percentage of water frozen is 88% in lamb, 91% in fish, and 93% in egg albumin. Living cells contain much water, often 2/3 of more of their weight. In this medium, there are organic and inorganic substances, including salts and sugars and acids in aqueous solutions, and more complex organic molecules such as protein, which are colloidal suspension. Thus, more the salt, sugar, minerals and proteins in a solution, the lower its freezing point, that is the depression of freezing point, and the longer it will take to freeze when put in freezing chamber. If water and fruit juice for example, are placed in a freezer, the water will freeze first. Further, unless the temperature is considerably below freezing point of pure water, the juice will never freeze completely, but rather will become icy and slushy. That is why most of the foods freeze at the temperatures below 0 to 3 degrees Celsius. The temperature of the food undergo freezing remains relatively constant until the food is mostly frozen, after which the temperature approaches that of freezing medium. The freezing process is thus a process of lowering of the product temperature from its original value to the storage temperature. From each point, the heat must be removed by conduction to the surface of food. The surface heat is then removed to the refrigeration medium. The freezing time depends upon number of factors such as dimensions, shape of product, thermal properties, temperature of refrigerant medium. Frozen foods have an excellent safety records and freezing has never been reported to be cause of food poisoning. The great advantage is that microorganisms do not grow in foods when the temperature is minus 10 degrees Celsius or colder. Foods preserved by means of other preservation method, chilling, drying, canning, have been more or less directly involved in food safety problems because these foods are stored at temperatures that allow microbial growth. Freezing has long been established as an freezing excellent method of preserving high quality in food products. Generally, freezing preserves taste, texture and nutritional value of foods better than any other preservation method. When done properly, it is the most satisfactory method of preserving many fruits and vegetables. Moreover, the original flavor, color, texture and nutritive value is usually retained during freezing in comparison to other methods of freezing. Freezing, however, does not add anything to the original qualities of fresh fruits and vegetables. Therefore, for the highest quality frozen foods, they must be of highest quality in their fresh state, and well-tested ways of preparing them for freezing must be carefully followed. Frozen foods are considered as healthy. US Food and Drug Administration (FDA) petition on March 25, 1998 approved an American frozen food industry petition to allow frozen produce to be labeled as healthy. The nutrient profile of selected raw fruits and vegetables and frozen of the same fruits and vegetables revealed relatively equivalent nutrient profiles. In fact, most fruits and vegetables during transportation or during retail storage undergo severe depletion of nutrients and essential vitamins. These losses can be prevented if they are frozen soon after they are harvested. There are some other terms which are often used while describing the aspects of low temperature storage and are loosely defined. The common terms are like cold storage, refrigeration and freezing. The cold storage, the temperature that is 16 to minus 20 degrees Celsius, whereas in the case of refrigeration 4.5 to 7 degrees Celsius. Freezing minus 2 degrees Celsius to lower. Freezing storage, it will be minus 18 degrees Celsius. In the cold storage, spoilage microorganisms can grow rapidly at temperature that is greater than 10 degrees Celsius. Some grow below 0 degrees Celsius if water is unfrozen. In the case of refrigeration, pathogenic microorganisms grow slowly, psychotropic microorganisms grow. In the case of freezing and storage, no significant growth of spoilage or pathogenic microorganisms. However, multiplication of microorganisms can take place if food is thawed. The advantages of frozen fruits and vegetables. The fresh vegetables and fruits closely resemble their frozen counterparts in freshness, since the metabolic activities are arrested to such an extent that all enzymes are inactivated and microorganisms are under control. The taste, flavor and color of fruits and vegetables are preserved to a maximum. They have high nutritive value since the retention of nutrient is maximum. Since frozen vegetables have already been subjected to a heat treatment, they require less time for cooking, thus saves considerable time in kitchen and also saves fuel. Greater convenience in handling and preparation. Freezing is a suitable choice for preserving fruit juices containing anthocyanin and carotenoid pigments, since the retention of pigment is maximum. They offer more hygienic food. 10% edible portion of food in each package. Since the degradative effect of heat treatment is bypassed in this method of freezing can retain the pigment of such fruit juices and concentration in the best form. Freezing can also serve as an intermittent technology for preserving commodities in bulk and supplying in a different form when demand arises. Example, bulk quantities and during demand, defrosted put in a brine solution packed in a flexible pouches and circulated in market whenever required. Value for money, especially during off season. No pollution problem in consuming areas. The waste collected during freezing can be utilized for production of value added products. Limitations. Some water soluble nutrients may be lost during freezing as the process involves blanching in boiling water. Users of quick frozen foods need to be invested on freezers to maintain the quality of the product till used. This adds to the cost of the product. Proper freezing transport facilities have to be developed for each product. Quick freezing can be handled at the industrial level because it involves specialized freezing equipment, technological know-how, strict quality control which would not be feasible on a small scale. Sales market have to be established in all markets with refrigerated display cabinets. The freezing point of a food may be described as the temperature at which a minute crystal of ice exists in equilibrium with the surrounding water. An increase in solute concentration during freezing causes change in pH, viscosity, surface tension, and redox potential of the unfrozen liquor. As the temperature falls, individual solutes reach saturation point and crystallize out. The temperature at which a crystal of an individual solute exists in equilibrium with unfrozen liquor and the ice is its eutectic temperature. Maximum ice crystal formation is not possible until this temperature is reached. Commercial fruits are not frozen to such low temperatures and unfrozen water is therefore always present. Rapid freezing causes the food surface to form a crust and prevents further expansion. This causes internal stress to build up in the food and makes pieces more susceptible to cracking or shattering, especially when they suffer impacts during passage through continuous freezers. During freezing, heat is conducted from the interior of a food to the surface and is removed by the freezing medium. The factors which influence the rate of heat transfer are the thermal conductivity of the food, the area of the food available for heat transfer, the distance that the heat must travel through the food, that is size of the pieces, temperature difference between the food and the freezing medium, insulating effect of the boundary film of air surrounding the food, packaging if present is an additional barrier to heat flow. It is difficult to define the freezing time precisely, but two approaches are taken. The effective freezing time measures the time that the food spends in a freezer and is used to calculate the throughput of a manufacturing process. Whereas the nominal freezing time can be used as an indicator of product damage as it takes no account of the initial conditions or the different rates of cooling at different points on the surface of the food. Freezers are broadly categorized into mechanical refrigerator and cryogenic freezers. Mechanical refrigerators which evaporate and compress a refrigerant in a continuous cycle and use cooled air, cooled liquid or cool surfaces to remove heat from foods. Cryogenic freezers which use solid or liquid carbon dioxide, liquid nitrogen or liquid freon directly in contact with the food. An alternative classification based on the rate of movement of the ice strength is slow freezers and sharp freezers, 0.2 cm per hour, including still air freezers and cold stores. Quick freezers, 0.5 to 3 cm per hour, including air blast and plate freezers. Rapid freezers, 5 to 10 cm per hour, including fluidized bed freezers. Ultra rapid freezers, 10 to 100 cm per hour, that is cryogenic freezers. All freezers are insulated with expanded polystyrene, polyurethane and other materials which have low thermal conductivity. freezing process, the product temperature is lowered and most water is transferred into ice crystals. With decreasing temperature, the liquid phase becomes more and more concentrated. As the volume of ice is about 10% larger than the volume of water, internal pressure of food may rise to 10 bar or 87 more or more. 10 bar or more. This pressure is sufficiently high to cause undesirable textural changes. Freezing textural changes freezing in foods but not as high as necessary to inactivate microorganisms. The size of ice crystal may vary with the method of freezing, that is quick freezing or slow freezing. Quick freezing is defined as the process where the temperature of the food passes through the zone of ice crystal formation in 10 minutes or less. The process removes quick removal of water and small ice crystals are formed in it. Whereas slow freezing involves slow removal of water and process may take 3 to 72 hours. This also involves ice crystal formation which have more damaging effect on the texture. The advantages of quick freezing over slow freezing are that smaller ice crystals are formed as there is less mechanical destruction of intact cells of the food. There is shorter period of solidification and therefore less time for diffusion of soluble materials and for separation of ice. There is more prompt prevention of microbial growth. There is more rapid slowdown of enzyme action. Quick frozen foods therefore are supposed to thaw to a condition more like that of an original food than slow frozen foods. Fruits and vegetable pieces which needs to be frozen must preserve their natural color, flavor, texture and be free from antioxidants and other additives. This is especially required for high moisture product ingredients such as fruits, salads or ice creams. Apart from maintaining sensory properties, they should also retain nutritive properties. Chemical and physical actions of freezing are highly detrimental to fruit and vegetables since their texture is mainly ensured by turgor pressure. Rupture of cell wall due to growth of ice crystals or enzymatic action during freezing causes loss of shape and prevents its return to its initial state. Loss of shape causes increased drip loss during thawing and less defined texture. Water is an important molecule involved in the deterioration reaction during freezing and freezing storage. Water within food migrates towards cold outer surface and then evaporates, recrystallizing as ice. Food surface becomes dry and color fades. Water may also participate directly in deterioration. Freezing reaction, which involves changes in color due to enzymatic or non-enzymatic reaction on pigments and production of off flavor. To minimize such changes in food products, it is important to use certain pre-freeze treatment prior to freezing. These treatments reduce the detrimental changes by inactivating the deterioration reactions and by reducing the water content in the material. Common pre-treatments used prior to freezing include washing, blanching, soaking and packaging. Blanching is a common heat treatment commonly used for most vegetables that have to be frozen and ultimately cooked before use. The aim of blanching is to inactivate enzymes responsible for deterioration of food. Inactivation is achieved by denaturing the proteins that would otherwise take part in reactions leading to deterioration. However, there are also certain disadvantages of blanching, like cellular tissues are affected by high temperature, there will be loss of texture and an increased risk of microbial contamination due to removal of food's natural and microbial flora. This can, however, be overcome by using improved blanching treatment employing high temperature short time exposure rather than low temperature long time. The different types of blanching methods are immersion, steam immersion. Immersion blanching refers to a blanching by directly immersing the food particles in boiling water. This treatment has number of advantages and disadvantages. Sometimes certain additives may be used in blanching water to complete blanching. For instance, addition of citric acid 0.5% to immersion bath decreases pH. Addition of bisulfite 0.5% prevents mushroom browning, and alloying of cauliflower. Blanching is quickly followed by a cooling stage. This is done by cooling the product under cool water to bring down its temperature. Rapid cooling avoids microbial growth on the exposed surface. Steam blanching by steaming has the advantage of minimizing the leaching out of soluble materials. However, at the same operating temperature, steam blanching takes 20 to 40% longer than the immersion blanching because of poor thermal exchange. Dehydrofreezing, sometimes depending upon the type of food and its final use of product, some moisture is removed from the food. This is partial dehydration. Partial dehydration is generally achieved by air drying. This process is called dehydrofreezing, that is dehydration prior to freezing. It offers many advantages over conventional freezing. Like during processing and preservation, saves energy, better quality and stability for partial air drying. Selection of food ingredients is very important. The food ingredient selector should be of high water activity. The water activity that should be greater than 0.98. Since water removal is limited to 50 to 60% of the original content. To avoid browning, blanching or other treatments such as dipping in antioxidant solution, ascorbic or citric acid, sulfur dioxide can be used. The air dehydration step usually produces a weight loss up to 50%. Apple, pear, peach and strawberry are commonly dried by air drying. Partial water removal from the foods lead to the concentration of the cytoplasmic components within the cell and to the depression of the freezing point. However, there are certain disadvantages associated with such drying. Free flow of the product is hindered due to particle agglomeration. Color is affected. For example, kiwi and strawberry fruits are susceptible to heat modification. For such fruits, air drying must be replaced by osmotic dehydration, which is more effective even at room temperature and which operates away from oxygen. Osmotic dehydration, conventional air drying can be substituted by or combined with osmotic dehydration as a pre-freezer treatment. The process involves placing the solid food, whole or in pieces, into solutions of high sugar or salt concentration. Usually, sucrose, cornstarch, syrup have been used as osmotic solutions. The principle behind osmotic dehydration is same as that of osmosis. Placing the food pieces in high osmotic solutions lead to osmosis and concentration of the cytoplasmic content. After pre-treatment, the material is subjected to the freezing step. Fruits and vegetables can be frozen by various methods. Cooled air freezers. In chest freezers, food is frozen in stationary, that is by natural circulation of air, at between 20 degrees Celsius and 30 degrees Celsius. The chest freezers are not used for commercial freezing owing to low temperature rates 3 to 72 hours, which result in poor process economics and loss of product quality. Cold stores is ice formation on floors, stores and ice formation in the floors, walls and evaporated coils caused by moisture from the air or from unpackaged products in the store. This condenses to water and freezes on the cold surface, which reduces the efficiency of the refrigeration plant. Uses up energy that would otherwise be used to cool the store, creates potential hazards from slippery working conditions and falling blocks of ice, and requires frequent defrosting of evaporated coils. In blast freezers, air is circulated over food at between minus 30 degrees Celsius and minus 40 degrees Celsius at velocity of 1.5 to 6.0 meter per second. The high air velocity reduces the thickness of boundary film surrounding the food and thus increases the surface heat transfer coefficient. In batch equipment, food is tucked on trays in rooms or cabinets. Airflow is either parallel or perpendicular to the food and is ducted to pass evenly over all food pieces. Blast freezing is relatively economically and highly flexible in that foods of different shapes and size can be frozen. Belt freezers or spiral freezers have continuous flexible mesh belt which is formed into spiral tires and carries food up to a refrigerated chamber, therefore self-stocking. This eliminates the need for support rails and improves the capacity by up to 50% for a given stack height. Cold air or sprays of liquid nitrogen are directed down through the belt stack in a counter current flow, which reduces weight losses due to evaporation of moisture. Fluidized bed freezers are modified blast freezers in which air at between -25°C and -35°C is passed at a high velocity 2 to 6 meters per second through a 2 to 13 centimeter bed of food contained on a perforated tray or conveyor belt. In some designs, there are two stages. After the initial rapid freezing in a shallow bed to produce an ice glaze on the surface of food, freezing is completed on a second belt in beds 10 to 15 cm deep. The formation of a glaze is useful for fruit pieces and other products to have a tendency to clump together. Food comes into greater contact with the air than in blast freezers, and all surfaces are frozen continuously, simultaneously and uniformly. These products, higher heat transfer coefficient, short freezing times, higher production rate and less dehydration of unpackaged foods than the blast freezing does. The equipment therefore needs less frequent defrosting. Cooled liquid freezers. In immersion freezers, packaged foods is passed through a batch of refrigerated polypropylene glycol, brine, glycerol or calcium chloride solution on a submerged mesh conveyor. In contrast with cryogenic freezing, the liquid remains fluid throughout the freezing operation and a change of state does not occur. The method has high rates of heat transfer and capital costs are relatively low. It is used commercially for concentrated orange juice in laminated card polyethylene cans and to pre-freeze film wrapped poultry before blast freezing. Cooled surface freezers. Plate freezers consist of a vertical or horizontal stock of hollow plates through which refrigerant is pumped at -40°C. There may be batched, semi-continuous or continuous in operation. Flat, relatively thin foods, for example filleted fish, fish fingers, are placed in single layers between the plates and a slight pressure is applied by moving the plates together. This improves the contact between the surfaces of the food and the plates and thereby increase the rate of heat transfer. Advantages of this type of equipment include good economy and space utilization, relatively low operating costs compared with other methods, little dehydration of the product and therefore minimum defrosting of the condensers, and high rates of heat transfer. The main disadvantages of the relatively high capital cost and the restrictions of the shape of the foods to those that are flat and relatively thin. Scraped surface freezers are used for liquid or semi-solid foods, for example ice creams. Freezing is very fast and up to 50% of the water is frozen within few seconds. This results in very small ice crystals which are not detectable in the mouth and thus gives a smooth, creamy consistency to the product. The temperature is reduced to between -4°C and -7°C and the frozen aerated mixture is then pumped into containers and freezing is completed in a hardening room. The cryogen is an intimate contact with the food and rapidly removes heat from all surface of the food to produce high heat transfer coefficients and rapid freezing. The two most common refrigerants are liquid nitrogen and solid or liquid carbon dioxide. Both liquid nitrogen and carbon dioxide refrigerants are colorless, odorless and inert. In liquid nitrogen freezers, packaged or unpackaged foods travel on a perforated belt through a tunnel where it is frozen by liquid nitrogen sprays and by gaseous nitrogen. The use of gaseous nitrogen reduces the thermal shock of the food and recirculation fans increases the rate of heat transfer. The main disadvantages is the relatively high cost of refrigerant. Liquid nitrogen is also used in spiral freezers instead of vapor decompression refrigerators. The advantages include higher rates of freezing and the smaller units for the same production rates because heat exchanger coils are not used. Continuous type fluidized bread freezers, particulate foods are fed by a shaker onto a porous straw. The food is pre-chilled and high velocity refrigerated air fluidizes the product, freezes it and moves it in a continuous flow for collection and packaging. An interesting feature of this unit is continuous and automatic defrosting. Air is blown via fan through cooling coils and up to the porous food trough. Cold air tends to condense its moisture onto cooling coils. A spray of polypropylene glycol antifreeze is maintained over cooling coils to melt ice as it would be formed. In this way, cooling coils are maintained at a maximum operating efficiency. Indirect contact freezing is based on the principle of placing the food on plates, trays, belt or cold walls so that it is in indirect contact with the cold wall. The food usually has flat packages, is placed between shelves and there is provision after loading for applying pressure to squeeze the shelves into more intimate contact with the top and bottom of the packages for faster freezing. All is enclosed within an insulated cabinet. The efficiency of freezing depends on the extent of contact between the plates and the food. For this reason, packages should be well filled or slightly overfilled to make good pressure contact with the plates. Immersion freezing with cryogenic liquids. Cryogenic liquids are liquefied gases of extremely low boiling point such as liquid nitrogen, liquid carbon dioxide with boiling point of -196 degrees Celsius and processing and -79 degrees Celsius respectively. Although liquid nitrogen is capable of freezing food down to -196 degrees Celsius, this is virtually never done because it entails unnecessary cost and could even be damaging to some foods. The food is seldom frozen to a temperature below -45 degrees Celsius and the quality results largely from the speed at which the temperature is reached. In most of the cases of fruits, vegetables, meat and fish items, it may take 1 to 3 minutes. Major advantages of liquid nitrogen freezing are such as it undergoes slow boiling at -196 degrees Celsius. This provides great driving force for heat transfer. Liquid nitrogen, like other immersion fluids, intimately contacts all portions of irregularly shaped foods, thus minimizing resistance to heat transfer. No primary refrigerant is required to cool this medium since low temperature results from the evaporation of liquid nitrogen. Liquid nitrogen is non-toxic and that to food constituents. Moreover, it can displace air from food and thus minimize oxidative changes during freezing and through packaged storage. High quality frozen foods are produced because of high speed of liquid nitrogen freezing. High cost of liquid nitrogen remains to be its major limitation. Liquid nitrogen is manufactured by compressing air and simultaneously removing the heat of compression. The cooled compressed air is then allowed to expand through specially designed valves. This expansion causes further chilling. The main effect of freezing of food quality is damage caused to cells by ice crystal growth. Food emulsions can be destabilized by freezing and proteins are sometimes precipitated from solution. Meats have more flexible fibrous structure which separates during freezing instead of breaking and the texture is not seriously damaged. In fruits and vegetables, the more rigid cell structure may be damaged by ice crystals. The extent of damage depends on the size of the crystals and hence on the rate of heat transfer. During slow freezing, ice crystals grow in intercellular spaces and deform and rupture adjacent cell walls. Ice crystals have a lower water vapor pressure than regions within the cells, and water therefore moves from cells to the growing crystals. Cells become dehydrated and permanently damaged by the increased solute concentration and a collapsed and deformed cell structure. On thawing, cells do not regain their original shape and turgidity. The food is softened and cellular material leaks out from the ruptured cells, which is termed as drip loss. In fast freezing, smaller ice crystals form within both cells and intercellular space. There is a little physical damage to cells, and water vapor pressure gradients are not formed. Hence, there is minimal dehydration of the cells. The texture of the food is thus retained to a greater extent. Concentration effects. It's seen that during freezing, concentration of solutes take place. Damage due to concentration results in gritty, sandy texture of food due to precipitation of solutes out of solution. Salting out of proteins effect due to solutes remaining in concentrated solution. There's drop in pH causing proteins to coagulate. Gases in solutions are also concentrated. This can cause supersaturation of gases and ultimately force them out of the solution. Frozen beer or soda pop may have such defect. Ice formation, that is nucleation and crystallization. The freezing process includes two main stages. First is the formation of ice crystals, which is termed as nucleation, and subsequently crystallization or increase in crystal size. Nucleation is nothing but combining of molecules into an ordered particle of sufficient size to serve as a site for further crystal growth. It is important for freezing to initiate. When the temperature falls below 0 degrees Celsius, viscosity increases and the nucleation stops. Now crystals start growing. It is important to understand the type of fluids in the cells. There are two types of fluids, extracellular fluid and intracellular fluid. The concentration of salts and other soluble products is higher within the cell than outside. The cell membrane acts as osmotic barrier and maintains the difference in concentration. When the product is frozen, the first ice crystals are formed outside the cells since the freezing point is higher. The cells lose water by diffusion through the membrane and the water will crystallize to ice on the freezing surface of the crystals already formed in the extracellular space. As the cells lose water, the remaining solution within the cells become more concentrated and their volumes shrink, causing the cell walls to collapse. The large ice crystals formed outside the cell wall occupy a larger volume than the corresponding amount of water and therefore will execute a physical pressure on the cell wall. When the pressure becomes high, it can damage the cell wall and lead to release of intracellular fluid. If the freezing rate is high, ice crystals are formed outside the cell. Only at a very high freezing rate, small crystals are formed uniformly throughout the tissue, both externally and internally with regard to the cells. Freeze cracking. Generally, light freezing rates lead to small ice crystals and to a better quality food systems. However, some products may crack when submitted to high freezing rates as in cryogenic fluids. This is particularly seen in mango slices frozen by liquid nitrogen. Pre-cooling prevents freeze cracking. Moisture loss or ice crystal evaporating from the surface area of a product produces freeze burn. This surface freeze-dried area is very likely to develop off flavors. Packaging in heavy weight, moisture-proof packaging material will prevent freezer burn. Drip loss. Formation of ice crystal causes a physical pressure on the cell wall. When the pressure becomes high, it can damage the cell wall and lead to release of intracellular fluid. This is called drip loss. Rancid flavors. Another group of chemical changes that can take place in frozen products is the development of rancid oxidative flavors through contact of the frozen product with air. This problem can be controlled by using a wrapping material which does not permit air to pass into the product. It is also advisable to remove as much air as possible from the freezer bag or container to reduce the amount of air in contact with the product. The winning strategy of frozen good industry is to continually improve quality. High quality raw materials containing high level of vitamins, especially water soluble ones, like vitamin C and minerals to ensure that after pre-treatment such as blanching, the nutritional value remains high. Color, flavor and texture are yet other factors that affect the quality. Peas is a good example of the cultivar effect and harvesting period on the quality of frozen product. Consumers and the frozen industry prefer sweet and tender varieties with small seeds, which are completely different from what is required by the canning industry and the fresh market. When the peas are ripening, their sweetness is due to sugar about 6% and low starch content 1%. Immediately after harvesting, there is a rapid reaction conversion of sugar into starch which can exceed 4%. This reaction determines a gradual reduction of sweetness, an increase in firmness and sensation in mealiness. Interval between harvesting and freezing fruits and vegetables undergo fast changes in chemical composition, sensory attributes and nutritional value. The product should be processed as quickly as possible. Example, peas and spinach have intense metabolism, high respiration rate after processing, which means severe decrease in quality if the time between harvesting and processing is too long. By using quick refrigeration, this negative phenomenon can be stopped. In general, the lower the temperature of frozen storage, the lower is the rate of microbiological and biochemical changes. However, freezing and frozen storage do not inactivate enzymes and have a variable effect on microorganisms. Relatively, high storage temperature between -4 degrees Celsius and -10 degrees Celsius have a greater lethal effect on microorganisms than do lower temperatures between -15 degrees Celsius to -30 degrees Celsius. Different types of microorganisms also vary in their resistance to low temperature. Vegetative cells of yeast, molds, and gram-negative bacteria, for example coliforms and salmonella species, are most easily destroyed. Gram-positive bacteria, for example Staphylococcus aureus and Enterococci, and mold spores are more resistant, and bacterial spores, especially Bacillus species and Clostridium species such as Clostridium botulinum, are virtually unaffected by low temperature. The majority of vegetables are therefore blanched to inactivate enzymes and reduce the number of contaminating microorganisms. In fruits, enzyme activity is controlled by the exclusion of oxygen, acidification, or treatment with sulfur dioxide. At normal frozen storage temperature, -18 degrees Celsius, there is a slow loss of quality owing to both chemical changes and in some foods enzymic activity. These changes are accelerated by the high concentration of solutes surrounding the ice crystals, the reduction in water activity, that is to 0.82 at -20 degrees Celsius in aqueous foods, and by changes in pH and redox potential. The main changes to frozen foods during storage are like degradation of pigments, chloroplast and chromoplast are broken down, and chlorophyll is slowly degraded to brown pheophytin even in blanched vegetables. In fruits, changes in pH due to precipitation of salts in concentrated solution change the color of anthocyanins. Loss of vitamins. Water-soluble vitamins, for example vitamin C and pantothenic acid, are lost at sub-freezing temperatures. Vitamin C losses are highly temperature dependent. A 10 degrees Celsius increase in temperature causes a 6-fold to 20-fold increase in the rate of vitamin C degradation in vegetables and a 30-fold to 70-fold increase in fruits. Loss of other vitamins are mainly due to drip loss, particularly in meat and fish. Residual enzyme activity. In vegetables which are inadequately blanched or in fruits, the most important loss of quality is due to polyphenol oxidase activity which causes browning, and lipooxygenase activity which produces off flavor and off odor from lipids and causes degradation of carotene. Proteolytic and lipolytic activity in meats may alter the texture and flavor over long storage periods. Oxidation of lipids. This reaction takes place slowly at -18 degrees Celsius and causes off odors and off flavor. Physical changes to ice crystal, for example, change in their shape, size and orientation, are collectively known as recrystallization and are an important cause of quality loss in some foods. There are three types of recrystallization in food. Isomers recrystallization. This is a change in surface shape or internal structure, usually resulting in a lower surface area to volume ratio. Accretive recrystallization. Two adjacent ice crystals join together to form a larger crystal and cause an overall reduction in the number of crystals in the food. Migratory recrystallization. This is an increase in the average size and reduction in the average number of crystals caused by growth of large crystals at the expense of smaller crystals. A wide variety of materials have been used for packaging of frozen foods, including plastics, metals, and paperboards. Polyethylene (PE), low-density polyethylene, and high-density polyethylene are commonly used for packaging of foods such as fruits and vegetables and fish. Materials are easy to seal and can be easily printed. LDPE is also easily available and cheap. HDPE can tolerate temperature in excess to 100 degrees Celsius. Both HDPE and LDPE, however, provide relatively poor barrier protection from oxygen. Polyester terephthalate (PET) is another common material used. The trace of the materials are suitable for reheating in conventional and microwave oven with stability at temperature in excess of 250 degrees Celsius. However, the materials are expensive and can become brittle at freezer temperature. Polystyrene (PS) is also a general plastic material used for frozen food applications. Commercial storage of frozen products is usually done in deep freezers at -18 to -20 degrees Celsius. Goodbye for today. Continue your studies with IIOCS.com Food Tech Club. For any queries, contact the number on the screen.